I don't quite know how to follow that, actually. It seemed like he was talking about somebody else. It's a great pleasure to be here and a great honor to be the scientist in residence. I've had a great time while I've been here. I can see many of the students in the audience, and now they're going to listen to me speak twice. Hopefully they weren't forced to come here, and they wanted a little bit to hear me speak, but it's great to see you again and other members of the audience. So I'd like to thank the university for arranging this, and everybody who's been involved in it. As I said, a wonderful visit for me. What I'm going to do in this lecture, which will be pitched as a lecture for the general public, will be to talk a little bit about the excitement of cancer research at the moment, about how we think this is exactly the right time to make progress. There's been some great technological advances and changes in the way that we think about cancer over the last few years. And we think this is what's going to make the difference. Now, there's still some way to go. The inroads we've made are small in terms of numbers, but the potential is absolutely enormous. And I have absolutely no doubt that when we look back in 10 or 20 years' time, we'll really see this as the golden age of cancer research, where we actually started to deliver therapies that work really well on people and don't make them really sick while we're curing them. And I think we do need to think about cure, not just keeping disease at bay or keeping people alive a bit longer. We should be aspiring to cure. So what I'll talk about first is that excitement and the science that underpins it, advances in genome technologies that have happened over the last few years. And then I'll talk about the history a little bit, about the history of cancer treatment, where it has come from, why we are where we are now. And then I'll talk about recent advances, new therapies, and then I'll finish up by talking a little about my own work in this area. And George has introduced it very nicely. And I will also now address the issue of why the Breakthrough Breast Cancer Charity started, because there was George asked why it was Breakthrough, and it was because the guy who founded it wanted it to do something meaningful, to actually make breakthroughs in breast cancer research. And the guy who founded it was called Bill Friedman, and he was a rather wealthy gentleman who had a wife who got sick from breast cancer at a relatively young age of early 40s, and rather quickly she developed advanced disease and died. And what he wanted to see was that other women wouldn't die in the same way that she died. And actually, I'll tell the story of what happened when I get to the end, because actually what he had hoped, in a strange way, when I first met him, it kind of translated into something meaningful. But I'll come back to that. So this is the summary of my talk. I'll talk about genetics and genes, and the history of cancer treatments, and then something about how cancer can be targeted, and then the future of cancer treatment. So let's start off with the basics. Familiar to almost everybody, I would suspect now, because it's become part of popular culture. So DNA is a code that has 4 letters that make up the code, T, C, G, and A. And the combinations of these letters make up genes, which is the individual hereditary unit. And a gene makes a protein, and proteins are what make us up. So there are about 20,000 of these in the genome, in the whole set of DNA, which of course is identical in every normal cell of our body. These genes are only about 1% of the total DNA, and the rest of it is a matter of some speculation, the dark matter of the genome that's now being uncovered. Now, the total size of the genome is really quite large. 3 billion letters make up the genome, and if you were to put that into a conventional book, it's 5,300 page books of continuous letters. And that specifies us. And what I'll show you in a moment is one letter difference in 3 billion can actually give you a very high risk of cancer. And that's what underpins cancer risk in some cases. And DNA varies. So here's a continuous stretch of a piece of DNA of that 3 billion. And one letter here has been changed. So that's what can be called a variation. We all have many, many variations. So some small percentage of our genome is different in every single one of us. In most cases, that is completely passive, has absolutely no effect. In some cases, what makes us look and behave differently and have a different risk of disease. Those small variations. Now, the ones that result in disease are often called mutations. So we have these variations, which we have millions of, mostly no change. But this is, if we determine your genome sequence, we would be able to tell a lot about you from those individual variations. You can tell about ethnicity. You can tell about restricted populations and disease risk. Even things like height, eye color, skin color can be got from the genome. And these mutations are generally, so the word mutation is a kind of prejudicial variation, if you like, that it is something that causes an alteration that is usually visible and perhaps disease-like. Now, the Human Genome Project, I'm sure you're all aware of this, and this is the project to uncover the list of letters and make that book, those 5,000 books of 300 pages. And this took 10 years to do one human genome and cost $3.7 billion. It was a major investment. It was said to be the equivalent of sending man to the moon. And it was incredibly important because it allowed us for the very first time to see the list of parts that make us up. It didn't tell us how we worked, but it told us what the limits were on what we contained. It was a list, a book, an encyclopedia that we could look things up in. But it was very costly. Now, technology to determine genome sequences evolved very rapidly. So just 5 years ago, we could do, using 10 people and about 74 of these type of machines, we could do about 1/30th of the human genome in about 4 weeks, if you really had a big genome center. Now, I have a machine on the bench in my laboratory where 2 people can generate the equivalent of 3 human genomes in 1 day. Right? So this has speeded up incredibly. And the cost has also plummeted. $3.7 billion for the first one. Very soon it will be on the order of $1,000, and I think it'll get a lot cheaper. And in fact, those of you who know anything about computers will know about Moore's law, which is the cost of a computing unit, goes down in a fairly predictable way like this. And this guy Moore came up with this idea early on. And most computers actually, when everybody changes their computer every 3 years because the thing just stops working because the software is too big, the storage capacity goes up every single time in a predictable way. The cost of sequencing, of determining genome sequences, is going down faster than the Moore's law predicts. So actually, what this means in practical terms is the cost of actually determining the sequence is now almost irrelevant. It's the cost of processing and storing the data that becomes really, really important, because data storage will be incredibly important when we sequence all your genes, all your genomes, which will happen probably within 10 years. We'll do everybody's genome at birth, and that will be held as a record, a lookup table of potential illnesses over your entire life. But the storage implications are really quite difficult to get our heads around at the moment. But I have absolutely no doubt they will be solved. If we can have an internet that has YouTube on it, YouTube is 1,000 petabytes. It takes 1,000 petabytes to store 1 million genomes. So if we can have something like YouTube, which is great, but, it's okay. I mean, it's a thing. but you've got 1 million genomes, right? So if we can afford YouTube, we can afford this. I think. You may think YouTube is better than 1 million genomes. I don't know. Some are looking skeptical at the back there. They obviously like YouTube. So how do we apply gene analysis to cancer? So we need to really think about what cancer is. And this is the great German pathologist Virchow, who really started the whole business of analyzing tumors by microscopy and classifying them by what they look like down a microscope. And until very recently, this is the standard classification method for cancer. Effectively, the tumor is removed either by surgery or biopsy. Slices are made in that tumor. They're placed on a microscope with some sort of stain, and that stain can give you an indication of what kind of cancer it is and maybe what the prognosis is. But the classification system related to where the tumor was and its rough characteristics. So it was really a definition, as I say, from the 19th century, which was when this method was invented. And of course, that has pertained up to only a few years ago. I employ, 25 pathologists in my institute who all day look at slices of tumor and then put some mysterious brown stuff on it and look down a microscope. It's not very high tech, right? This is really very basic stuff. So we need to be moving away from this to understand the molecular makeup of cancer in order to make proper classifications based on the genes. Now, a very important point that's emerged over the last few years that actually has been really an underlying theme for many years is that a particular cancer type, like breast cancer, is not a single type of cancer. It's probably 40, 50, 100 different types. And these are the types that are visible down the microscope. So using this 19th century technique, you can distinguish different types of cancer, breast cancer. And I can just about tell you that that's lobular by looking at it. These things here, you need to be a professional to diagnose them. And in some senses, they are useful because they give a rough idea of how that tumor might behave. But they give no insight into what the genes that are altered in that cancer or how we might treat it. So we're moving away from this type of classification into a much more sophisticated methodology. And really that's based on this understanding of our genes, these 3,000 million letters, and analysis of cancer genomes. So a cancer arises in a normal cell of your body. That's why it's difficult to treat, but it becomes mutated over time. And recent studies suggest that there may be 20,000 to 100,000 genetic changes in every cancer. So out of 3 billion, that doesn't seem like very many. But if these are involved in making a cancer, that's really quite a lot. So cancers are very damaged normal cells. This is what the genome looks like when you paint the chromosomes with a dye that tells you which chromosome is which. This is supposed to be chromosome 1 here, which is supposed to be 2 copies of a yellow chromosome. You have 2 copies of every chromosome in the normal cells. There's 4 in this box, but Lord knows what that is. I mean, that's a mixture of 7 or 8 different chromosomes that have become rearranged and scrambled. So really this gives you a sense of what the problem is here. This is a cell that looks and behaves like a normal cell, except it keeps growing. But its genome has become deranged, and because of that derangement, has the ability to do things that normal cells normally don't do, like grow in places they shouldn't grow. They don't die when they're supposed to die, and they grow very fast. So that's the challenge, is to understand what that genome sequence of the cancer genome is telling us. So what I showed you before was the old histological picture of breast cancer with things that you can see down the microscope and the classifications you make using the 19th century technique. This is the 21st century equivalent. These are maps of breast cancer genomes revealing the rearrangements that are available when you look at the genome. And what you can see here is And each line represents an interchange between a chromosome. So the more lines, the more rearrangements. And what you can see here is a gigantic range of things going on here. This thing looks almost normal. It's a breast cancer. This thing looks incredibly deranged. That happens to be a BRCA1 mutant breast cancer. Very deranged, but huge variety. So now the challenge is to use this to classify real information, digital information about the genome, the 3,000 million letters, and use that to come up with a classification system to inform us about the relationships between different cancers and to give us thoughts about how we might treat it. So how do we use this information for cancer therapy? So I'll just have a few slides on the history of cancer treatment, and it really is a fascinating subject about how we got where we are. But it's important to recognize that now, at the moment, cancer is treated by what are essentially medieval kind of torture chamber tactics. You cut the tumor out with a big knife or a small knife, and most people who are cured of cancer are cured by surgeons. Or you take what is a quite sophisticated way of burning people, a radiotherapy machine, and it burns the last remaining cells around a tumor. And again, many patients are cured by radiotherapy, blunt tool that it is. And finally, you can poison the cancer. When I say poison, I'm not exaggerating. These are genuinely poisons. If they were in your kitchen, you would put them right at the top shelf at the back because you do not want to touch these things because they are genuine poisons. And just to demonstrate that, I'll show you how the first chemotherapies were derived. So in the First World War, both sides used poison gas extensively. It was used as a tool to deny areas to the enemy, largely, and to disable soldiers. And it was these burns, actually, that were so useful as a weapon because a soldier with a burn like that simply couldn't fight. And, of course, it killed a lot of soldiers. And many of the early poison gases were things like chlorine gas. But later on, and I think it was the British that invented mustard gases, and they have this characteristic kind of structure that you'll see in a few different things. They have these chlorines on the end and some molecule in the middle. They're called mustard gases. And what these do is that they are very poisonous. And the first clue that these might actually be useful as cancer therapies were actually published in 1919. But we're looking at autopsies of soldiers that had died of poison gases and noticed changes in the bone marrow to suggest that bone marrow cells were killed. And this kind of bubbled under for a while, and nobody really gave it serious consideration until later on, until, in fact, the Second World War, whereas I think everybody knows that poison gases weren't used in the Second World War. Both sides feared massive retaliation and the consequences of using it. Of course, it was against the Geneva Convention. But, in fact, the Americans took steps to, in case the other side used poison gas, to have a lot ready. But they did this in total secrecy. And during 1943, the Americans had transported, actually to the British, 100 tons of mustard gas, the same stuff, to Bari Harbor, which then was bombed, and a lot of soldiers were exposed to mustard gas. So this wasn't a kind of an attempt to use it, this was an accident, but it was suppressed at the time for actually almost 20 years, because Winston Churchill didn't want it to look like the Allies had been preparing to use mustard gas just because the potential consequences. And importantly, there was a lot of clinical investigation of the effects of mustard gas on the survivors. And what they saw was huge shrinkage of lymph nodes and of white blood cells. And that information was transmitted secretly to the US and gave a great stimulus to the use of these mustard gases in cancer therapy. And they were actually used first for lymphoma and leukemias because of the link to bone marrow and to lymph nodes. And here, this was the very first chemotherapy, mustine, and you can see it's got this wiggly bit, 2 chlorines, and this has got a nitrogen. And if I just go back quickly, that looks almost identical to that, right? So the poison gas became the chemotherapy that was used first, with a very small chemical modification. So we are literally poisoning people with these agents. This drug was made in my institute in all 3 of these drugs, in fact, were made in the building I work in now, in my institute, in the early 1950s. All 3, in fact, are still used today as poisons to treat cancer in patients. So, I mean, when I talk about this in context for our institute, I never know whether to be proud of it or ashamed of it, actually, that 60 years later, we're still using things that were developed in the First World War to kill people, but we're using it to treat people, to poison them. But they still work, actually, in a certain context, but with horrible side effects. So that's the basis of cancer chemotherapy, right? Poisoning. And then more modern cancer drugs have developed by finding toxic chemicals in, for example, the Pacific U, which was then purified to this molecule Taxol, which is a famous cancer drug. Again, incredibly toxic, but managed use can give some results as chemotherapy. But notice it took this long to get approved. Huge time delays because of difficulties in manufacture and because it's really toxic. So these types of chemotherapies, relatively non-specific effects. What they're targeting are the hallmarks of cancer, like rapid replication, that you actually, that when you try and kill cells that rapidly replicate, you have effects like bone marrow toxicity, gut toxicity, and hair loss, because you're effectively targeting rapidly growing cells, and that's all chemotherapies do. They're not selective. And there's also a difference in the response in different cancers. There are some cancers that are cured and don't recur by chemotherapy. Testicular cancer is a brilliant example of that. Lance Armstrong was cured of testicular cancer, even though he had cancer in his brain and his liver and his lungs. It is because it's exquisitely chemosensitive. So that's a small subgroup. There's a variable response cancers like breast cancer, where a small fraction of people are cured by chemotherapy. But for every 10 women that receive chemotherapy, cytotoxic chemotherapy, only 1 benefits. So that's a kind of an alarming statistic, that we are massively overtreating with chemotherapy, but we don't know which one is the one that's going to respond, so we have to treat 10. And then there's cancers for which modern cancer researchers should be ashamed, like pancreatic cancer and lung cancer, where we have made no impact whatsoever in survival over the last 20 to 30 years. The biggest advance in the treatment of pancreatic cancer, drug treatment of pancreatic cancer, has been the use of a drug called gemcitabine, and that provides a 4-week survival advantage over the other treatments. So this is dreadful. Right? So we are not going to do any better with standard chemotherapies. So we need an alternative approach. And thankfully, that's what the genome advances are providing. Insights that we can use to use this information, understand the genomes, to provide targeted therapies that can hit the cancer much harder without touching the normal cells. That's the principle, because cancer is different, genetically different, so let's treat it with something that attacks the cancer but doesn't make people sick. Now, I need to just tell you 1 or 2 technicalities about what kinds of genetic changes there are in cancer. So there's 2 main changes that affect cancer development, critical mutations. So of the 20 to 100,000 genetic changes, there may be 10 that are really important. And we have 2 kinds of genetic changes, what are called oncogenic changes, and they effectively act as accelerators. So it's as if the accelerator on a car has been flawed, and it causes unrestrained cell division. These are often present in cancers, so that you get this accelerated growth. And then to compensate for that, to allow it, there's also what's called tumor suppressors. And they are effectively as brakes on cell division, And they are often lost in cancer. So it's this double effect of having the accelerator down, the brake pedal is inactivated, and that's what causes this massive uncontrolled growth. So the challenge is, how can we attack the accelerators and reimpose the brakes so that we can slow down the cancer so that it doesn't become a problem? And I want to give you a couple of examples before moving to my own work. And the first is a rare cancer of the stomach called gastrointestinal stromal tumor, called GIST. And this is a picture from an endoscope of a GIST. And it's traditionally very resistant to chemotherapy, and it's almost always lethal. And 50% of these tumors turn out to have a mutation in a specific gene. Here, in this particular T residue, it's shifted into, I think, a G residue. So one change here is critically important. One change out of 3,000 million is very, very important. That's what drives this cancer. And how it drives it is this gene makes a protein that sits on the cell surface. It's called KIT. And KIT is activated by a molecule that binds to it, and that then signals, normally it signals controlled cell proliferation. And when you get the mutation in this gene, you get suddenly uncontrolled cell proliferation by having an activation that shouldn't happen. It's an oncogene change, the accelerator is down. What has been developed is a drug called imatinib, also called Gleevec, that binds to this mutant form of this receptor and blocks it. And what that does is stop the accelerator. And that drug was developed for this specific purpose of blocking this particular action. And this is what causes tumor cell death when you add this drug. And this just shows the dramatic effects of using this drug on a gastrointestinal tumor. These are patients treated with the drug, and this is with standard treatment. And you can see here the overall survival, 50%, is about 6 months. Half the people are dead. With this new treatment, most of them are still alive at 2 years. So a really dramatic effect. This drug was also developed for a leukemia called chronic myelogenous leukemia. And this is the effect on the disease over time. This is the incidence of the disease. The incidence, so the same number of cases are present now as were present when the drug came in 1997. But what you can see now is that death from this disease is now a fraction, a very small fraction, of what it was in 1997. So a massive effect on this disease. Now, this isn't a common disease, but if you've got it, this is the drug you want to have. And it's a great approach. And a similar thing has happened with another lethal disease, melanoma. Again, there's a gene that's mutated called BRAF. In this disease, melanoma, that depending on the state which you get it, can be a very nasty disease and very difficult to treat. Chemotherapy doesn't work very well. A drug was developed called vemurafenib that binds to this gene and causes these really dramatic effects. This is a scan here, and you can see these are lesions of melanoma spread throughout the body. Look at this absolutely dramatic effect. And I've met some patients who are on this study. They know actually that this is not going to cure them, but what they are think is incredibly valuable is the year it gives them of extra life to do the things that they wanted to do in that extra year of life with actually feeling quite good. And many of them were very debilitated by this disease, and suddenly they feel able to do a lot of things. So an incredibly valuable development. Now, in moving on to my own work, the question arises, that's great for these accelerators, but how do you target these brakes? And the problem with doing this is intrinsic to these types of changes. But first I have to tell you a little about hereditary predisposition to cancer, because that provides a context for understanding what we're trying to achieve. So small numbers of cancers are the result of a strong hereditary predisposition. And I think we're all kind of familiar with families where there's a lot of cancers. And over the last, in fact, this has been recognized now for about 70 or 80 years, that probably breast cancers and ovary cancers, in some cases, ran in families. But it was only more recently that this became amenable to analysis. And you can see in this family, you've got female breast cancer and male breast cancer. You also get ovary cancer in these families. And so identifying the gene that predisposed it to this has been a big effort. And as George said, my involvement in the identification of the BRCA2 gene is, I suppose, one of the proudest things, the things I'm most proud of, because it really changed the way that people were treated. So the story was that we identified this gene, and it's a rather, I haven't got time to tell the full story, but it was a rather dramatic story of a race between our academic lab with not much resource and a large-ish biotech company in the US that were trying to identify this gene in order to patent it. And in the end, we won and we lost. We won the race to find the gene. We patented it first so that we could gift it to everybody to test for free. But the American company also patented the gene, and after us, in fact. But in the US system, you have to go to court very, very, very expensively in order to fight such an action, and we weren't able to do that. So that has resulted in testing in the US being controlled by Myriad. But in Europe, they haven't really bothered to sue us. Well, not yet anyway, although they keep threatening it every now and again. So we are free to test, and we gave a free license to anybody who asked in Europe to test for this gene. But why this story was particularly dramatic was, first of all, it was incredibly rapid, the whole thing. And things were happening overnight. So it was 10 days from us sending a paper for publication in Nature, for review in Nature, to the paper being published in print, on paper, pre-electronic. So 10 whole days from review to printing, which was kind of strange. But also happening at the same time is we were starting to test people. This woman was scheduled for surgery at the Royal Marsden Hospital, our sister hospital, and she was for prophylactic mastectomy. So she had a family history, like the family I've just shown you, and had decided to have her breast removed before she got cancer so that she wouldn't die of cancer. And she'd done that consciously, and she was set for surgery, and the week before the surgery, we were able to use the new discovery of the gene to test her. We found the mutation in her family, but she was negative. So immediately we saved that woman from a disfiguring operation. But to her, most importantly, her two daughters could not have inherited that mutation from her because she didn't have it. So this just shows the absolute power of the genetics, the power of this kind of discovery. And, even if it was only one woman, it was worth it. But, of course, it's been now used on thousands of women who've made informed decisions about their lives because some were told they didn't have the gene, some were told they did have the gene, and some have surgery to remove the affected tissue after careful consideration and counselling. So I think that showed the impact. So these genes have a very high risk of breast and ovary cancer and some other cancers. Carriers have an up to 85% risk of breast and about a 40% risk of breast cancer. And why do they get breast and ovary cancer? Well, what we know is they have a mutation in this gene, BRCA1 or BRCA2. So one change out of 3,000 million bases. And they have, in the normal tissues, they have it in every cell of their body, this mutation, but they also have one normal copy. So we have two copies of more or less every gene in the genome, if you're a woman. And so what happens in the tumor, we worked out quickly, was the normal copy is lost. So it's the normal copy that protects cells from becoming tumors. When this is lost, it becomes a tumor. And the effect, so genes make proteins the effect on the protein of having this mutation is to cause it to be truncated. It's smaller and it doesn't function very well. So what you've got in tumours is this non-functional bit, and normal cells in the carriers have this functional bit. So the question was, for many years, after we discovered this, how do you target something that's absent? How do we kill a cell because it's got a lack of function rather than a gain of function? I've shown you how cells that acquired a function might be killed with drugs. How would you do this? And that's where we used genetics. And we also spent, like many labs, trying to understand why the cancer arose in the first place. And the reason is to do with the repair of your genome. Now, it turns out your genome, your normal genome, is actually really quite a fragile molecule. It breaks quite frequently, both spontaneously and about 10 times higher if you go out in the sun, for example. And it's not just a few breaks. There's all sorts of different kinds of breaks. And you will suffer, whether you like it or not, about 1 million billion individual episodes of DNA damage during this talk, depending on how long I talk for, obviously. The way I'm going, it'll be 1 billion billion, but I'll speed up. So what we've all evolved, is a mechanism to repair these things, because with that level of damage, with no repair, we just fall apart. And so this evolved very early in the evolution of life, the ability to repair your genome. Now, when you lose BRCA1 and BRCA2 function, when you've got a mutation in the tumor, you lose the ability to keep your genome intact properly. So they have a specialized function in DNA repair, probably when the double helix breaks on both strands. And this specialized form of repair is lost in these mutation carriers. Now, if this meant the cell died and couldn't maintain its genome at all, that would be a good thing. But that doesn't happen. What it does is use other mechanisms that aren't as good to repair its DNA, so it does it in a very sloppy fashion. So the upshot of why cancers arise in mutation carriers is that normal damage is badly repaired. And when it badly repairs, it causes the scrambling of the genome, and then that causes the activation of all sorts of inappropriate genes, and those inappropriate activations are the things that cause cancer. And this just shows what a tumor genome looks like from a BRCA mutation carrier. I used this as an illustration before, but you can see it's incredibly scrambled. Now we get on to the kind of conceptual bit. What we used was a concept that had been developed originally in the 1920s by a scientist called Bridges, who worked in the fruit fly, and then given a name by the scientist Theodor Dobzhansky in 1946. And it's a genetic concept used to understand how genes interact with each other in fruit flies. It's really quite arcane, but it's actually quite easy to understand once you kind of get your head around it. So I'll take this bit a bit slowly, but I've got an illustration in a second that I think helps. So synthetic lethality is an idea that if you imagine two genes that are in a genome, and we've got 20,000 genes, but these are just two particular genes, A and B. If you remove gene B by making a mutation, in this situation, nothing is happening. If you remove gene A on its own, nothing happens. But sometimes when you remove these two things together, the cell dies unexpectedly. And why might that be? Well, I think the analogy to an electrical circuit is quite good here, where if you imagine circuits in parallel, the removal of one, you can use the other parallel arm. And the same with this one, but you remove both and the cell dies. So it's a backup pathway, if you think about that. So A normally backs up B, B backs up A, you remove both and you've got nowhere to go. So it's a genetic concept. So we started to think about how to use that to kill cells that lacked BRCA function and couldn't repair their DNA properly. So as I said, tumors that have this deficiency in BRCA1 have a specialized absence of a DNA repair pathway. What we're going to do is to block another pathway, pathway B, and then the aim is to get on normal cells, pathway A can rescue, but in the mutant cells, pathway A and B are eliminated, and the idea is to get lethality here, death to the tumor cell, but life to the normal cell. So it sounds all very complicated, and here's a more straightforward explanation, I suppose, that some people like and some people don't. So we imagine that these genes are legs of a table. What we're going to do is knock away one leg of a table, and a kind of three-legged table is a bit wobbly, but it can stand up. Whereas if you pre-remove one of the legs, so that's a BRCA mutant cancer, it can't repair DNA. Then when we come in and kick one of the legs away, the whole table falls over. So it's that double whammy effect here. Stability here, well, relative stability, but lack of stability here, causing specific tumor cell killing. So what we did was we came up with an idea of kicking one of these table legs away using a drug. And what was amazing is when we put this drug on the cells that had the BRCA mutation, what we saw was this incredible effect. So these are normal, these are normal cells that have a BRCA mutation. So they're tumor cells from a BRCA mutation carrier, just in the normal state. Now when we add this drug that inhibits this pathway that kicks the leg away, you can see these chromosomes look, sorry, these are chromosomes, not cells. The chromosomes look very fuzzy and unresolved. And that's because they are dissolved, in fact. So they cannot maintain the genome at all when you add this drug. So this drug called a PARP inhibitor. And so you can see that that concept played out in a massive kill with this drug, killing an absence of function, a lack of DNA repair. So what we were able to do was to take that observation directly into the clinic. And what was very exciting was the clinical trial of this agent started just a few months after we published our original observation. And the reason was the drug had been in development, but it wasn't clear who you were going to treat or how you were going to use it. So what we were able to do was to design the clinical trial. The clinical trial was only performed on BRCA mutation carriers. And what we saw within 6 months of starting the trial was some pretty spectacular responses in patients that were essentially resistant to all known therapies. So of 70 patients treated in the trial, more than 50% had a very strong clinical response. Now bear in mind that these patients had everything that modern medicine could throw at them. They were really quite ill, life expectancy of 3 months or less. And multiple patients were still alive after 2 years. So there was a huge difference, and one could see it on scans. They felt better, their tumor burden had gone down, and many of them lived for several years, even though they were in a very, very advanced state. And I think what was remarkable about this, again, it was one of these things where just by chance and by being in the right place, something translated very rapidly from laboratory to clinic. This just shows some scans from these patients, showing the quite remarkable responses. This is a patient with breast cancer. This mass here was really quite large, very considerably reduced here. This is an ovary cancer patient. Again, this mass has disappeared. And both these patients went on to be on treatment for 2.5 or 3 years. So the question is, are there more Achilles heels in the genome? And I think there are many, and I think we can start to exploit similar ideas to treat cancers in this kind of way of attacking weaknesses and defining strengths, and then looking at genetic interactions. And this thing here that looks like some planets from a distant universe is a genetic interaction map of yeast. It's starting to understand how all the genes in yeast play with all the other genes in yeast. Yeast is a very simple organism to study. Human cells are much more complicated, but I think we can start to develop insights like this into human cells, which means that we can do what we did with the BRCA mutant situation in a systematic fashion by, first of all, understanding the cancer genome of individual patients, understanding how those genetic interactions interact, and then making the best of the various treatments, so that we move away from cancer management being related to looking down a microscope and a pathologist with years of experience can tell a slight difference in the way the cancer just simply looks, to a situation where we have proper biomarkers of who might respond, and we can select exactly the right therapy. So the principles of this is the therapies we're developing will not work on everybody. They cannot work on everybody because not everybody has the same tumor. We need to define the tumor that you have, not the person sitting next to you has, and give you the right treatment and not give it to the other person. That's the important principle. So existing cancer therapies have been designed on a one-size-fits-all principle. How can we get the best for the average patient? There is no such thing. And that's why the success of chemotherapy, though very valuable in some situations, is ultimately limited. And if we move to targeted agents, understanding these things, I think we're going to make great inroads into better patient selection using biology, the principles of biology, the principles of genetics, and a rational approach rather than simply something that happened to be an extract of tree discovered in South America that happens to kill cells and kill tumor cells slightly better than it kills normal cells. So I think we are in this age now. This is the future, if you like. The genome revolution is already here. Now it's about implementing this, I think. And I think that's why there's a great sense of excitement. A young medical oncologist yesterday said he thought it was brilliant that he was just starting his career now. And he wouldn't have wanted to start it 20 years ago because he's now got his future in front of him. Is this brilliant science combined with these fantastic advances in medicine. And he can see that things are changing. And I think there's a general feeling of that really things are. So I think we are in this kind of golden age. There's a lot of issues that need to be resolved. I mentioned the kind of data issues that need to be challenged, but I think we'll get there. It's still early days for genomic medicine. We've only had the genome. So we've been doing the thing with pathology for 150 years. We've had the genome for less than 10 years. And already it's started to make inroads. So when people say, that genome, it didn't really do anything, did it? My God, come off it. We've only been doing it for a few years. Give us a break. So there are difficulties. The pharmaceutical industry also, we're reliant on the pharmaceutical industry to make drugs, make therapies, because it's unachievable in academia because of the high costs. We can do the early bit, we can inform them what to do, but actually delivering drugs, we need a strong pharmaceutical industry that's well informed by biology, and that's not always the case. But with those reservations, I think, yes, it is now this golden age. And I just end by talking about the guy who founded Breakthrough Breast Cancer, Bill Friedman, his wife, who was called Toby Robbins, and I became director, and I knew Bill Friedman, and I knew his motivation that his wife had died of cancer. But he stopped being involved in the charity because the research center was started by the charity Breakthrough Breast Cancer, which he'd founded. So I became director of the center in 1999, and then he really stopped being involved in the center, and I didn't see him very much. And then towards, I think 2008 or 2009, I had lunch with him, and he told me something I had never known, and I don't know why I never knew this, but his wife was a BRCA mutation carrier, and of course then the penny dropped. I'd been hired to run the research center to make breakthroughs, and so that his wife, what had happened to his wife wouldn't happen to other women. And what it ended up doing, by coincidence actually, was we developed a therapy that could have been used on his wife, and if used early enough, might have saved her life or at least extended it. So in a weird way, what he set out to do, somebody says, right, I'm going to build a research center and I'm going to make a cure for my wife's cancer. You know, that's the stuff of films. Nobody ever says that. Nobody ever believes that that's a reality. But that's actually what happened. I'd like to think I'd intended it, but whatever. The outcome was quite interesting. I'm delighted to entertain any questions whatsoever, and thank you very much for your attention and for this great honour. No, if there's any technical questions, I'll answer them in a non-technical way.
It's not working, I think. No, it's working. Yeah, thanks, Alan, for this fantastic talk, and I think it gives a great sense of the fascination that discovery can bring to the scientist, and of the fascination we have now that we see this dramatic change in translation of findings from in basic research to the clinics that is now on a good way. So now, thanks again, and I would like to have questions from the audience. Okay.
I wonder what the other 99% of the genome are doing when someone has cancer.
Yeah.
The dark matter, so to speak.
So that I think that's a very interesting issue. So we don't know what 99% of it does, but 99% of it contains most of the mutations. So there are clearly ways of affecting how genes work by making mutations in that dark matter. We know that. So there are mysterious ways in which the genome is regulated, and that's just being uncovered. But it probably is some of it is collateral damage. So because cancers take probably 20 years to develop, that cell has to go through a lot of replication and making itself, and it makes errors each time. So there's the damage that occurs, and I think what is very interesting we're learning from the genome sequences is that cancer has recorded the entire life history of the individual. So if it's a lung cancer in a smoker, by analyzing the genome sequence, you can tell how many cigarettes that person smoked in their lifetime, more or less. So there's a direct correlation between number of cigarettes and strength of cigarettes and the number of mutations. Well, you may even get brand, actually. Well, brands are being banned anyway, aren't they? So they're all in just anonymous boxes now. The amount of tar, you could certainly tell. So the same with UV exposure, because that leaves a record. I suspect there's a record of a lot of other things in there. Certainly the number of cell cycles, the DNA repair capacity, and maybe causes of cancer that we don't even anticipate at the moment. Will be revealed by genome analysis because we don't know what the cause of most breast cancers are, for example. So that might be revealed.
So my question relates to the point that you made, the important point that you made, that certain cancers need certain treatments. I wonder why carriers of the BRCA mutation develop breast cancer.
Interesting
question. And why the PARP inhibitor then doesn't kill or only kills the cancer cells and not any other cells that also carry those mutations in the body.
So it's not clear why BRCA mutation carriers mainly get breast and ovary cancer, but they actually do get a higher risk of, men get a higher risk of prostate cancer. Both men and women have a higher risk of pancreatic cancer as well, and male breast cancer. But it's predominantly breast and ovary. And I've been asked questions about this now since 1995, and I'm none the wiser as to the answer. But I think it's something to do with the environment of the breast and ovary. They are both hormone responsive. They both cycle during the menstrual cycle. The breast proliferates, the ovary follicle breaks through the surface of the ovary, and the epithelium is repaired on a cyclical way. And it may be because it cycles, somehow the BRCA mutation is allowed to develop in that context. But the answer to the PARP inhibitor thing, if you've got a BRCA mutation and it causes pancreatic cancer, that pancreatic cancer responds to PARP inhibitor, which is really quite remarkable given what pancreatic cancer normally responds to, which is nothing. It does actually respond, and there's been a few patients treated. So it's to do with the gene and not the site of the tumor, and I think that's one incredibly important principle about everything I've said. It's the genetics that matter, not where the cancer is. And where the cancer is can affect the genetics, but the genetics is primary.
My question sort of follows the one made by Hemo. You showed us the nice picture of this concept of synthetic lethality with a table and one leg for BRCA, one leg for PARP. But for sure, in the cancer cells, the BRCA leg is off. You add the PARP leg, and the tumor or the table falls down. But so in the same time, you kick off the first leg of the healthy, of the normal cells, the first leg of the table, the PARP leg. So how can you exclude doing nasty things to those cells?
Yeah, because it's only got 3 legs and it's wobbly. And I think that's why it's not a bad analogy. We don't know what's happening in the normal cells when you inhibit PARP, but the toxicity is relatively minor. So these are patients treated for 2 or 3 years. Maybe in the long run, one would induce some sort of other malignancy because it's going to block a DNA repair pathway. And sometimes people are concerned about that, and then I immediately point out that doctors who treat cancer have been causing cancer for many, many years by giving things like nitrogen mustards that cause leukemia. And there is a perceptible increase in cancer patients. That may sound like a dumb thing to say because they've already got cancer and obviously they're cancer prone. But it's clear that it's induced by the treatment. So although it's a theoretical risk, we think it's a relatively low risk. And if I had the choice between a lethal cancer that I might have an extension of life with and then I get a leukemia in 10 years' time, I'd hope that leukemia then would be curable. So it's, you want it now or after you're dead?
You showed us nicely that you have the concept with the accelerators and with the brakes, and that we can already nicely target the accelerators and we start to target the brakes. But cancer has a large plasticity, and you get resistance. Would you think that there's a difference in resistance development whether you target the accelerator or the brake?
Yeah, I think that's a very interesting question, and I'm not sure I know for sure what the answer is. But, I mean, just to generalize it, I think we're recognizing that cancer is a very diverse disease and is behaving by principles of Darwinian evolution. And, of course, how Darwinian evolution operates is by having a diverse population, that there's variation in a population. And then when there's some catastrophic change in the environment, and in this case that would be drug treatment, then what you get is selection of a variation and survival of the fittest. So what that means is there's variation in the cancer population. You kill most of them with a drug. One, for whatever reason, is resistant, and then that grows out and becomes the species, if you like. And I think that analogy is really good. It has its limitations because the time frame is obviously different. It's not billions of years. It's years. But I think we're starting to think that that's the difference. Now, what add on to the top of that, the drug itself is causing mutation. The chemotherapy is causing additional mutations and probably making the matter worse because you've got even more diversity. But it really speaks to the principle of cancer therapy that metastatic disease, where it's spread around the body, is always going to be a challenge to treat because there are many, many cells. The more cells, the more diversity, the more chance of resistance. Whereas if it's in an early stage and it's been treated by surgery and just killing a few cells, the diversity is so much less, and then by Darwinian principles, it's less likely that resistance will emerge.
Mm-hmm. I think there was a question.
I think what I read recently is that in the light of the human genome, we now notice that terminally differentiated cells have quite different genomes. So a liver cell is genomically different from a lung cell or whatever, epithelial cell. So these cells are really, really different. They have duplications and deletions that are normal for these cell types.
Yeah.
So this will hugely increase, I think, the picture you look at. So
yeah.
You would not have to sequence the human ones, but for every tissue. So you think there is a unifying mechanisms that you can apply, or would it be really necessary to look at every single tissue itself?
Well, I think what we'll move to is a situation where we're just analysing bits of the genome that we know are relevant to that disease, I think, and that requires an understanding of knowing what's relevant, and we don't know that at the moment. Adding to your picture of the normal DNA varying, and I think we don't quite know what the implications of that are yet, but certainly it does occur, is the variation within a tumour and how many genomes are present within a tumour that are different. And this comes back to the resistance idea. It may be that cancers arise like a branching tree, that they're all related through some sort of trunk, but actually within each cancer you've got things that have separated. And so which one is the cancer genome you're trying to treat is at the moment quite tricky. So I think you'll move to a situation, you're not treating the genome, you're treating the individual mutations, and that's what matters. And so the mutations are quite easy to spot because they're usually things that cause either a gain of function or a catastrophic loss, and so they're less susceptible to the interpretation of normal DNA. But it's not a solved problem. So it's, if you think, you go, well, it cost $3.7 billion to do one genome, and now we're talking about doing multiple genomes in individuals, then even no matter what cost it is, it's going to be a problem.
Yeah.
What do you think about the potential of small RNAs?
Yeah, so this also relates to, for therapy or for, so I think there's been exploration of kind of genetic solutions to this over several years. We thought about gene therapy. Now we're thinking about small RNAs as a potential drug to modulate genes. The moment the delivery issues, I think, are the critical thing, that it's very hard to think how you deliver something to every tumor cell in the body, especially with metastatic disease where it's spread about. And so how you would get it there, I think, is an unsolved problem. But if you could get it there, I think there could be some efficacy. And there's certainly quite a few people exploring this.
Now, since the future is now, where would you put your money in terms of tools? What tools are you thinking they are the most promising from the tools that we know they're available or they've been developed?
I think we've got a lot of technology and we need to configure it for the clinical situation rather than the other way around. So a lot of clinical development is driven because core facilities and analysis is done in a certain way, so it's kind of adapted. What we need to go back to is design clinical studies to answer a question, and then the technology adapts to that. So that's where I think that's a tool. Having the ability to deliver genome sequences is all very well, but if it takes 4 weeks, which is amazing, really, 4 weeks. It took 13 years. But 4 weeks is not good enough, because if you think about a patient that's got advanced disease and is really ill, and they've come off one therapy, is resistant to one therapy, and you want to shift to another therapy, you need 4 days. You don't need 4 weeks. That's still a very significant challenge. So I'm investing quite a bit of our institute resources into try and configure facilities so that they can deliver on clinical questions in a reasonable time frame. And I think imaging is also another one that's really quite important. So we've got the genomic piece. We need to be able to see cancers in situ. So PET, advanced PET tracers, I think, are going to be very important. And we also need a way, and this was at the symposium yesterday, of monitoring tumour growth and development without invasive procedures. So we need blood-based markers of whether the tumour is responding to treatment or is continuing to grow. And so I think I'd put my money behind that as well, as a way of a non-invasive way of rapidly assessing how the patient is doing.
And shouldn't we, so you showed us nicely that the breast cancer is highly diverse if you look on the genome. And when you compare the genomes of the different cancers, shouldn't we come to another picture that we group more to the pathways and not to the entities?
Yeah, I mean, I don't think we're quite at the resolution yet to be able to tell one cancer from another. And I think there'll be more overlap than we think. We know that the cancers of smokers are more mutated than the cancers of non-smokers, both of which are lung cancers. Mm-hmm. So the cancers of lung cancer sufferers who don't smoke, have never smoked, are actually really quite benign compared to the ones that have smoked. So that's a kind of dead giveaway. But apart from that, it's not absolutely clear. And one sees great diversity in things like colorectal cancer, where there can be 100-fold difference in the mutation frequency between two individual cancers, and really no clue why. But we need to understand the landscape. So we've called this the genomic scar. And what I mean by that is that there's a lot of damage in the genome that we don't know what's happened. So this history of its life, if you like, is recorded in its genes. So understanding that scar of its life, I think, can give us insights into where it came from, what it is, and then how to treat it. But that's just kind of an idea at the moment. It's not really kind of very well formulated.
Okay, so if there are no more questions, thanks again. Thanks also for the great discussion. And I will now hand over to our rector to give the award.
Yeah. We have a diploma for you, an award, of course, to show or give proof that you have been here. And it's my pleasure to read it to the audience. Die Universität Duisburg-Essen verleiht Herrn Professor Alan Ashworth, PhD, in Anerkennung seiner Leistung die Auszeichnung Scientist in Residence für seine Verdienste auf dem Gebiet der Tumorforschung, zu seinen größten Errungenschaften zählt dabei die Identifizierung von Genen, die für die Krebsentstehung, insbesondere von Brustkrebs, verantwortlich sind. Durch seine bahnbrechenden Forschungsergebnisse hat er in den letzten Jahren den Weg zur Entwicklung neuer und innovativer Therapien von Krebserkrankungen bereitet. Duisburg-Essen, am 7. Dezember 2012. Herzlichen Glückwunsch. John.